why smooth muscle lacks striations. Okay, what did we say causes the striation in skeletal muscle? What structure? The sarcomere, correct, correct. So the sarcomere is what gives skeletal muscle its striated appearance. Okay, and as a result of smooth muscle not having any sarcomeres, it has no striations, black striations, hence the term smooth. Now, some examples of where smooth muscle is found, we talked about this quite a bit already. Internal organs, your gastrointestinal organ, your stomach, blood vessels all throughout the body have smooth muscle in their wall, in their lining. The respiratory tract, the uterus, the bladder, you can go on and on. Right? Smooth muscle is found in a lot of places throughout the body, and it's not under our voluntary control. Okay? It's controlled by the autonomic nervous system, and so we do not voluntarily control blood vessels or respiratory tract and that sort of thing. Okay? Now, the shape of a smooth muscle cell is a spindle shape, right? Unlike cardiac muscle, unlike skeletal muscle. You don't have this straight parallel sarcomeres the way we do that type of muscle. And so all we have is that rounded, sort of spindle, elongated shaped cell. Now they're much smaller than skeletal muscle cells, again, because of that difference in organization of these cells and the lack of striations. Smooth muscle has dense bodies. We'll talk more about what dense bodies do, but they're unique to smooth muscle. Okay? And so just to point out some of those differences here, we have the striations apparent on skeletal muscle as well as cardiac muscle. Okay? Whereas in smooth muscle, we simply have these elongated spindle-shaped cells, which are much smaller and usually connected by gap junctions and dense bodies. Now, although the structure of smooth muscle cells differs greatly from that of skeletal muscle, we still have the sliding filament mechanism. Right, so that same cross-bridge cyclin and the actin and myosin filament that slide past one another, that same process pretty much still holds true for smooth muscle. Okay, but there's some slight differences that I'll point out. First of all, the actin and myosin are not in parallel, right? If we remember the skeletal muscle, the sarcomere, we had actin, myosin in parallel to one another. In smooth muscle, because this is a elongated spindle-shaped cell, we have the thick and thin filament in a more oblique arrangement throughout this cell. So they're not parallel as the way we see them in the sarcomere, they're more obliquely arranged, okay? And so the orange part here is indicating the thick filament, which is the myosin head, and then the longer thin filament is this blue stripe, which is your actin. Okay. Now some differences between the sliding filament in skeletal muscle and smooth muscle. In smooth muscle, the myosin head has to move across the entire length of the thin filament. Okay, and so what happens when these two filaments slide past each other is the thick filament slides to the opposite end of the thin filament. And so we have this crumpling of the cell. Okay, this crumpling of the cell. We also have dense bodies. So the dense bodies are these points of attachment where the thin, thick filament and the outer laying connective tissue is all connected to one. So at the dense body, the exterior connective tissue connects to the internal filaments, thick and thin filament. And this brings about synchronized muscle contraction, and it helps to spread that contractile force throughout the entire cell very quickly. Okay. Another important feature that we see here with smooth muscle is that there's a longer range of contraction. It's going to be a slower contraction. Smooth muscle contraction is not built for speed the way that skeletal muscle contraction is. Okay, so you have a longer, slower contraction for smooth muscle. Okay. Let's talk about the steps of excitation, contraction, coupling in smooth muscle. okay? Oops. So we have our smooth muscle cell, which we know is a spindle-shaped cell. So this is our smooth muscle cell. And then within that cell, we also have, very importantly, the endoplasmic reticulum. What is the function of the endoplasmic reticulum? Anyone?
Stores calcium.
Stores calcium, perfect. So the storage of calcium is in the ER. I got lots of calcium here, but then we also have calcium in the extracellular environment. So there's calcium outside of the cell as well, okay? Now there's a voltage-gated calcium channel on the membrane of the cell, as well as on the membrane of the ER. And so the first step in smooth muscle contraction is that we have a depolarization of this smooth muscle membrane. Right, so the membrane is depolarized, very similar to the end plate potential in skeletal muscle at the neuromuscular junction. And then that pretty much opens up these voltage-gated calcium channels, and calcium rushes into this cell. Right, so we got some calcium coming in, which brings us to step 2. Step 2 is called calcium-induced calcium release. Which basically means that when we open up this first voltage-gated calcium channel and calcium floods the cytoplasm initially, this triggers an even further response by opening up the voltage-gated channels on the ER, which further increases the calcium concentration in the cell. So there's more calcium rushing in. Alrighty? Now the function of that calcium brings us to step 3. Calcium basically binds to a molecule which is called calmodulin. Right, so we have this molecule called calmodulin. And when calcium binds to this molecule, we get what's called the calcium calmodulin complex. And all that that means is that calcium has bound to this molecule and activated it or energized it, and now it's ready to go and do its job. Okay, that brings us to step 4, right? What is the job of this calcium calmodulin complex? Well, it's gonna go and activate an enzyme. Now, let's talk about this enzyme. So this enzyme is called MLCK, and that stands for myosin light chain kinase. Now, this is an enzyme, and it's ending with kinase. What do we think is the job of this enzyme? What is this enzyme going to do in its activated state? Not break something down. So, no, it's not going to add calcium. It's going to add something. But we spoke about kinases and covalent regulation earlier in the semester. So what does a kinase do? What does a kinase do? I think you're on the right path, Ariel. I think I hear the right words.
No, like it's just like phosphatase. It was adding a phosphate. So is it gonna add? I feel like it's gonna add something.
Okay, so you have a little mix up. So a kinase is gonna add a phosphate, and then a phosphatase is gonna take away a phosphate, correct. So that myosin light chain kinase is an enzyme that's basically going to phosphorylate the myosin head in this sliding filament model. So now it's gonna be turned on or activated by this calcium calmodulin complex, and now it's energized to go and phosphorylate the myosin head. Now, so let's draw this here, right? So we can have phosphorylation of the myosin head. which basically brings about cross-bridge cycling. And just like what we saw with skeletal muscle, that brings about the shortening or the crumpling in this case of the smooth muscle cell, and that's muscle contraction. Okay, let's think about what happens in the instance where this muscle is relaxing. Okay, now MLCK is going to compete with another enzyme, right? And as we just mentioned, this other enzyme is a phosphatase. Okay, so if it's a phosphatase, what did we say it's going to do? So kinases add a phosphate group and phosphatases remove them, right? So if we have phosphatases dominating here, we're going to have dephosphorylation of the myosin head. And that is going to stop cross-bridge cycling. Which basically leads to muscle relaxation. Okay? And so what we really want to understand here is that depending on the level of calcium, just the way calcium was crucial to skeletal muscle contraction, it's also crucial here. But the important differences are, one, it's going to come from two sources. So in skeletal muscle contraction, we had one source of calcium. What was that one source of calcium? We had one source of calcium in skeletal muscle contraction. What was that source of calcium? So not smooth muscle, but think back to skeletal muscle. The calcium that brought about the cross-bridge cycling by attaching the troponin, that whole mechanism, where did that calcium come from? SR. The SR, good. So the sarcoplasmic reticulum. In the case of smooth muscle, calcium is not only coming from the ER, right, which is the same structure for smooth muscle, but it's also coming from the extracellular fluid. All right, so that's one important difference. The other difference is we have this regulation by these two enzymes. So calcium comes in, it activates this calcium calmodulin complex, and depending on whether we have excess calcium, if we do have excess calcium, we're going to activate MLCK, and if we don't have excess calcium, phosphatases will be activated. They'll go and grab those phosphate groups off of the myosin head and cease muscle contraction. Okay. All right. So in that way, calcium essentially regulates or turns on, turns off smooth muscle contraction, similar to the way it did in skeletal muscle.